Have you ever just stopped to think about how a single cell, something so simple, pulls off one of the most incredible tricks in all of biology? How it grows and then, with almost unbelievable precision, splits perfectly in two? It's this tiny microscopic engineering puzzle, and it's happening trillions of times a day, all over the world.
So let's boil it down to a simple question. How does a cell find its own belly button? Seriously, how does it pinpoint that exact middle spot to divide? It doesn't have a brain, it doesn't have eyes, there's no tiny cellular ruler. And yet, the accuracy it needs is just staggering.
And you've got to understand, for something like a bacterium, this isn't just about being neat and tidy. No, if it gets this wrong, it's absolutely catastrophic. I mean, if a cell splits off center, one of the new cells might not get any DNA at all. And that's a death sentence, plain and simple. So the stakes, they literally couldn't be higher.
So how in the world does the cell solve this life or death puzzle? Well, the answer is kind of counterintuitive, but it's also incredibly elegant. See, the cell doesn't actually search for the right place to divide. Instead, it just blocks off all the wrong places. It creates these no-go zones.
This whole unbelievably complex process really just comes down to two simple rules. Rule number one, and this is the big one, do not, under any circumstances, slice through your own genetic blueprint. And rule number two is, don't divide way out at the tips of the cell, what scientists call the poles. And there are two different molecular systems working together to make sure these rules are followed. To protect the DNA, the cell uses a process called nucleoid occlusion, and to create those no-build zones at the ends, it uses something called the MinC system. And what you're seeing here is the whole strategy in one picture. The nucleoid occlusion system creates that red forbidden zone right over the DNA. Then, the MinC system creates those green forbidden zones at the poles. So, what's left, right? Just that one little sliver of space, highlighted in yellow, smack dab in the middle. It becomes the perfect spot to divide, simply by default.
Okay, so let's dig into that first rule. Let's look at the clever little system that guards the cell's most precious cargo. The official name for this is nucleoid occlusion. And really, the name says it all. It occludes, or blocks, the whole area where the cell keeps its chromosome. It makes sure that the division machinery can't even get started there.
Now, the hero of this story is a protein called SLUM-A. You can think of it like a molecular guardian. It physically latches onto the DNA, and just by being there, it sends out this powerful do not build here signal. Wherever SLUM-A is hanging out, the parts for the division ring just can't come together. All right, so the DNA is safe. Check. Now, what about that second rule? How does the cell stop itself from dividing at the tips? Well, that is the job of the MinC system. And it's basically this dynamic trio of proteins. First, you've got MinC. He's the main bad guy, the inhibitor. Then there's MinD, who's like an anchor, holding MinC to the wall of the cell. And finally, you have MinE, who's the chaser, always keeping the other two on the move. The real muscle here is MinC. Its entire job is to stop construction. It's like a microscopic saboteur. It actively hunts down the very first building blocks of the division machinery and just breaks them apart.
But here's where it gets really amazing. These MinC proteins, they don't just sit still. As you can see here, they're in a constant, elegant dance, zipping from one end of the cell to the other and back again. And because they're always moving, it means the inhibitor, MinC, spends most of its time at the poles and almost no time in the middle. It creates this kind of molecular force field that's weakest right in the cell's center. And right here, you can see that sabotage in action. Those green bundles, those are the building blocks. And look, you can see the MinC proteins in purple literally wedging themselves in there and busting those bundles apart. It completely stops the division ring from ever getting started.
So let's put it all together. The DNA is cordoned off, the poles are declared no-build zones. The cell has just done something really remarkable. It's cleared one single perfect spot for division to begin. Those no-go zones are firmly in place. And just by a simple process of elimination, the middle of the cell is the one and only place left that's free from all those inhibitors. It's the only place where construction is even possible.
So as soon as the coast is clear, the first builder arrives on the scene. It's a protein called FtsZ. And with no inhibitors to stop them, these FtsZ proteins start linking up, forming this beautiful, perfect circle right at the midpoint. We call it the Z ring. Now, the Z ring is just the beginning. It's just the scaffolding, really, the big glowing sign that says, "Build here." And immediately, a whole army of other proteins rushes to the site, assembling into this huge, complex machine called the divosome. This is the actual construction crew that's going to build the new wall.
And you can see how specialized this crew is. You've got FtsZ, which we met, laying down that foundational ring. Then you have proteins like ZipA that act like clamps, locking the whole thing to the cell membrane. And then you have the real workers, like FtsI, who are the enzymes that actually build the new cell wall, brick by brick.
So let's just take a breath for a second and appreciate how unbelievably elegant this whole solution is.
Think about it. The cell doesn't do some crazy math problem. It doesn't measure anything. It achieves this perfect life or death precision just by saying no. Two simple don'ts create one perfect do, right in the middle, every single time.
And it all comes down to this simple idea. The cell doesn't need a ruler to find its center. It just needs to know where it's not allowed to divide. By simply blocking off all the wrong places, the one right place just reveals itself. It's an absolute masterpiece of efficiency.
It really makes you wonder, doesn't it? If there's this much genius, this much elegant engineering packed into the simple act of a bacterium splitting in two, what other incredible secrets are hidden in the unseen world, humming with life all around us?